Welcome to today's Drug Hunter Flash Talk. We're back after a somewhat longer spring break, and I'm your host, Dennis Koester. We're super happy you're all here today. And yeah, today we'll be talking about sulfoximines with the expert Ulrich Lücking himself. So welcome to the show, Uli.
Thanks. Hi, nice to see you, Dennis. Thanks a lot for the invitation. And yeah, thanks a lot, everybody, for dialing in.
Now, without further ado, I want to introduce Uli. Ulrich Lücking, he's the head of chemistry at Forks Therapeutics in Basel, although he resides in Berlin at the moment. And he has more than 20 years of experience in small molecule research. He was a project team leader at Bayer and Schering, and his efforts were instrumental in the discovery of 6 clinical candidates, including the CDK9 inhibitor inotuziclib and the ATR inhibitor ilimusertib. At Forks Therapeutics, he led the chemistry activity to the clinical candidate, Forks 428. That's a highly potent and selective PARC inhibitor with best-in-class potential. And over the years, Uli's research really contributed to the exploration of novel chemical space. For instance, the application of sulfoximines that he's going to talk about today, but also sulfone amides and some other macrocycles and other functional groups and innovative clinical candidates in drug discovery. He contributed to more than 75 patent applications and published more than 25 scientific papers. Prior to joining industry, Uli studied chemistry at the University of Hanover with a stint in Stephen Lay's lab at the University of Cambridge in the UK. And he completed his PhD under the supervision of Andreas Falz at the Max-Planck-Institut für Kohlenforschung in Mülheim an der Ruhr, and then carried out postdoctoral training with Julius Rebeck at the Scripps Research Institute in La Jolla, California. Well, welcome to the show, Uli. We're certainly super glad to have you today, super excited. We just talked about how you've already been working on sulfoxamines, for about 20 years. So that is super exciting to learn. And yeah, as usual, we want to kind of start with a few warm-up questions. So maybe if you could elaborate a little bit on what inspired you to start a career in drug discovery.
Mm-hmm. Yeah, so I think the decision to study chemistry in the first place was still kind of triggered by my interest in fireworks as a kid and as a teen. However, the decision to go into medicinal chemistry was, I think, influenced by my postdoc in the group of Julius Rebeck. So during my PhD, I worked on the optimization of a palladium catalyzed coupling reaction and its application to rather simple natural products. However, during the postdoc, I worked on so-called container molecules. So these were large vase-like molecules that were supposed to capture smaller molecules to study host-guest interactions. And the way of working was that, first of all, you would design such a container molecule with certain properties in mind. Then there was the synthesis, and if successful, you would not put the molecule on the shelf, but investigate the properties, for instance, by NMR experiments. And I, during that time, really enjoyed this design, make, and test cycle. And I also, realized that I like to design molecules, which is still what I'm doing today.
Yeah, amazing. Yeah, a lot of us, I guess, got started in childhood with fireworks and chemistry kits. Don't we all love that as Germans? And
Indeed, yeah.
You just mentioned something about molecular design, and I mean, I love to follow your post on LinkedIn, and I see so many kind of fascinating molecules. So I wonder, what's your favorite molecule and why?
Yeah. Okay, that's not that easy. So to decide, so I give you, let's say, my current top 3, just very briefly. So number 1 has to be Fox 428, our novel PARG inhibitor, which is about to enter clinical trials. So PARG inhibition, could really offer novel treatment options to cancer patients, and we believe we identified the best-in-class molecule. Moreover, it has been very exciting for me to see this molecule progressing all the way to clinical trials after joining a new startup as number 3 on the payroll. a couple of years ago. So then number two, is another Fox molecule. So surprise, but I will show this one later today in the presentation. So this is a covalent inverter inhibitor. And we employed a novel warhead for this molecule, or this series of molecules. I've been actually thinking about this warhead for probably 8 to 9 years, but never had the right opportunity to utilize it before in a program. And now it's exciting to see that it translates into a differentiated profile. And then the last one, going back to my interest in macrocycles. So I'm very impressed with the RAS inhibitors from Revolution Medicines. So they have several clinical candidates. I like especially, I think it's 9805. So this is a complex macrocycle, covalent mode of action, spiral spacer, employing a novel warhead. So this checks a lot of boxes for me. And I also think it's very impressive from the CMC side that molecules of this complexity can be brought to patients nowadays. So I think, this really all exemplifies the progress which has been made in the field.
Yeah, absolutely. No, all great molecules. Yeah, and I'm very excited to see, for me, for the first time, the Forks burner inhibitor today. So I'm really excited. What are you most excited about in drug discovery today?
So I think there are a lot of exciting things, and it seems to accelerate all the time, right? So first of all, I've focused pretty much on small molecules so far, but in the wider sense, and I think it's exciting to see that the lines between the different modalities become more and more blurry, and that small molecules still play a significant role. So I've been interested for a long time in the expansion of the chemical space, and I guess the sulfoximines are a good example. I think the expansion of the chemical space also initially triggered my interest in covalent inhibition. So this was more driven from the chemistry side. These sometimes weird-looking novel warheads coming in to tackle different types of amino acids raised my interest in the first place. But of course, covalent inhibition is also an intriguing concept with respect to pharmacology. And then, I have also long-standing interest in macrocycles. I synthesized muscone during my PhD, which was kind of the starting point. But also in industry, I started designing macrocyclic kinase inhibitors already in the first program. So at the beginning of the 2000s, and that's a topic I'm still very much interested in.
Yeah, super cool. No, a lot of exciting development and new modalities happening for sure. Yeah. So if you could kind of go back to the beginning of your career, Uli, and give yourself some career advice, what would you tell yourself? Your younger self?
I guess my main advice would be to reserve some time on a weekly basis simply for reading. So I guess sounds trivial, but it's often not that easy in the daily routine of a pharma job. So on the one hand side, I would recommend to get on track with DMPK and pharmacology early on. This is something that I probably neglected a bit in the beginning. So I was very much focused on the chemistry, which I guess is not a bad thing for a medicinal chemist. But still, I think if you understand PK and pharmacology better, then chances are that you can also design better molecules. And then I would also, really recommend to look into case studies and, for instance, also novel patent applications on a regular basis, simply to absorb different strategies or functional groups that you can use in your own programs to overcome optimization hurdles.
Oh, awesome. Yeah, great advice. Thank you, Uli. And then, last but not least question, what do you like most about Drug Hunter?
So I mean, we discussed it already before, right? So I mean, I already followed Dennis as a let's say influencer when he was still with Genentech. So this is exciting to see this emerging into a novel company and a growing company. So what I like is that it's driven by people with long-term industry experience. So it's on the one hand side, yeah, scientific, of course, but also quite entertaining. So I like the molecules of the month, molecule of the year. I'm also a regular user of the amount calculator for in vivo testing. Last but not least, going back to the recommendation to spend time with case studies, I also like the flash talks. So I think this is a very convenient format to stay on track about what's going on in the industry.
Awesome. Yeah, no, that's great. Thank you so much, Oli, for all this positive feedback. So I'll show like 1 minute. I wanted to quickly show you the series of reviews that we now actually have been writing. So we've been writing a series on bioisosteres. We've split that up into a non-aromatic and aromatic part, and the non-aromatic part is even split in 2 pieces. So you can find the latest here. Comes with an introduction. We talk about hydrogen isosteres. So here, for example, deuterium or fluoride. Highlight a lot of interesting case studies here in the field that are super exciting. Of course, sulfonamide bioisosteres, and a beautiful example here from Bayer that Oli was involved with is also documented here. And then a beautiful NLRP3 inhibitor from Novartis here. For a sulfone imidamide bioisostere, which is also really cool. And then a number of other carbonyl group bioisosteres are listed and documented here as well. So I highly recommend to check out the full review on Drug Hunter. And now, without further ado, I will hand it off to Oli for his presentation on new advancements on sulfoximines in medicinal chemistry. Oli, stage is yours.
Yeah. Thanks a lot again for the introduction and for dialing in. So let's see if I can get the pointer. Okay, no. Okay, so you can see the presentation in the pointer?
Yeah, everything looks perfect.
Okay, great. Okay, so then, I will talk about sulfoximines in medicinal chemistry. So I will get started with the program that got me interested in sulfoximines in the first place. And this was the pan-CDK inhibitor approach, which resulted in the identification of the clinical candidate roniciclib. However, in the main part of the presentation, I will highlight more recent examples of sulfoximines in drug discovery. So on the one hand side, I will show the recent clinical candidates, which contain a sulfoximine group. Interestingly, with only one exception, all of these compounds are NH sulfoximines. However, I think the utilization of the sulfoximine nitrogen position still is a neglected opportunity in drug discovery. And therefore, I will show several examples for the utilization of this position before I come to the summary. Okay, so at the beginning of the 2000s, at the former Sharing AG, we selected the potent pan-CDK inhibitor ZK304-709 for clinical evaluation. However, phase 1 dose escalation study in patients was finally terminated due to dose-limited absorption at high doses, which was mainly attributed to the limited aqueous solubility of 709. Moreover, the compound was found to accumulate in erythrocytes of patients due to an off-target activity against carbonic anhydrases, which was mediated by the sulfonamide group. So this off-target activity was also discussed as a parameter that contributed to the high interpatient variability of exposure that we recorded in patients. So to overcome the limitations of our first clinical candidates in the follow-up program, we applied the following strategies. With respect to dose-limited absorption at high doses, we wanted to increase the aqueous solubility, and we wanted to reduce the required therapeutic dose by increasing cellular potency. Moreover, we wanted to eliminate the off-target activity against carbonic anhydrases. Since the IP space in the aminopyrimidine series was very limited, we also considered a rather unusual suggestion at that time, which was to exchange the sulfonamide for a sulfoximine. This idea went back to a coincidental spotting of the excellent sulfoximine review by Regaline. So at that time, I had never heard of a sulfoximine group before, but of course, there was a certain optical resemblance of the sulfoximine group with sulfonamides, and the described properties in the review got me interested. So sulfoximines are isoelectronic with sulfones, but the introduction of the nitrogen creates a symmetry and offers an additional point for substitution, which can be utilized to further tune the properties of the sulfoximine group. In contrast to sulfones, the mildly basic nitrogen allows metal ion coordination or salt formation. Both heteroatoms bound to the sulfur are hydrogen bond acceptors, like in sulfones. But in case of NH sulfoximines, so when R3 equals hydrogen, the group has dual H donor, H acceptor functionality. Moreover, simple sulfoximines were known to be readily soluble in protic solvents. So overall, very interesting properties, and therefore we decided to indeed investigate the switch from the sulfonamides to the sulfoximine in our series, even though there was hardly any precedence of the use of the sulfoximine group in drug discovery at that time. So we prepared a simple model compound, which then revealed biochemical and cellular activity, but no activity against carbonic anhydrases. Moreover, the model compound displayed high hydrolytic and metabolic stability, which had been major concerns in our discussions before about utilizing this unusual moiety in our series. So the belief was that the sulfoximine group might be either hydrolyzed or metabolized right away to give the corresponding sulfone, but this was not supported by the actual data. Therefore, further lead optimization efforts finally led to the identification of roniteclib, which was the most potent pan-CDK inhibitor of our approach. And this significantly increased cellular potency in combination with a very good PK led to a 50-fold reduction of the required therapeutic dose in animal models. In addition, roniteclib revealed a significantly increased aqueous solubility, which was around about 20-fold higher than that of our first clinical candidate. The sulfoximine roniteclib did not inhibit carbonic anhydrases, and in line with our hypothesis, did not accumulate in erythrocytes anymore. Roniteclib then also revealed very promising PK upon oral administration in humans in phase 1, but the compound was stopped in phase 2 due to a safety signal. I think it's fair to say that the safety signal most likely did not go back to the sulfoximine moiety, but rather to the mode of action of roniteclib. None of our various competitors at that time was able to bring a pan-CDK inhibitor to the market neither. The initial success of roniteclib, however, triggered the evaluation of the sulfoximine group across projects at Bayer, and also outside of Bayer, sulfoximines have indeed become much more popular. This claim is supported by the results of a recent SciFinder substructure search. So you see here the number of patent applications which claim and exemplify sulfoximine compounds over the course of time. After its late discovery in 1949, the sulfoximine group did not create a lot of excitement in the life sciences for decades, but this started to change, let's say around 2010, and recently interest in the sulfoximine group is at an all-time high. I think an important driver for this development is the significant evolution of the synthetic methodology to make sulfoximine compounds. And you see here just, let's say, a personal selection. So the traditional method to make sulfoximines, which was used for decades, was the conversion of a sulfoxide to sulfoximine by the use of hydrozoic acid generated in situ from sodium azide and concentrated sulfuric acid. And in principle, this is a good method. We utilized it for more than a year in the pan-CDK inhibitor approach, and usually recorded very clean reactions, but the method, of course, has a severe safety limitation. So hydrozoic acid is not only very toxic, but also potentially explosive. And therefore, we basically performed the reactions only on 10 milligram scale. Therefore, I think the introduction of the rhodium catalyzed method from the Bohm group in 2004, which converts a sulfoxide to sulfoximine without safety issues, is a real landmark in the field. We also employed this method for the synthesis of roniciclib, and at that time it was very important for us to demonstrate that we would be able to support a clinical trial with roniciclib without any safety issues in the CMC part of the activities. However, I think lately many people like to use the one-pot method, which was first published by Bull and Louisi. So this directly converts a thioether to an NH-sulfoximine without safety issues, simply by stirring with ammonium carbamate and bis-acetoxyiodobenzene. There's a recent patent application which shows that the amounts of bis-acetoxyiodobenzene can be even reduced to catalytic when employing electrochemistry. Moreover, there's a paper from last year which shows that the method is suitable for on-DNA synthesis, which will hopefully lead to an increase of sulfoximine structures in their libraries. Moreover, we've seen the introduction of bench-stable reagents, which allow the synthesis of sulfoximines. So the Willis group in 2020, for instance, introduced this reagent, which offers the synthesis of sulfoximines and sulfonimides. Along those lines, Lobchuk last year introduced the SCARO reagent, which also offers the synthesis of sulfoximines and sulfonimides with high EE. So the increased interest and the improved methodology also contributed to a significantly increased commercial availability of sulfoximine building blocks. So when we started to look into the sulfoximines in the pan-CDK inhibitor approach, there were literally a handful of compounds that you could buy commercially, and this has really changed significantly. However, I think the main driver for the increased interest are actually case studies, and therefore I will proceed with a very nice example from AstraZeneca. It's the clinical ATR inhibitor ceralasertib, which is now in phase 3 clinical trials. So starting from the screening hit, initial lead optimization efforts led to the identification of AZ20, which is a potent and selective ATR inhibitor. However, concerns about a low maximum absorbable dose, going back to the low aqueous solubility, as well as a potential ZIP-TDI liability of the compound, triggered further lead optimization efforts, which finally resulted in the identification of the sulfoximine ceralasertib. So the compound shows comparable potency and selectivity, but significantly increased aqueous solubility and reduced log-D. Moreover, the switch from AZ20 removed to ceralasertib removed the potential ZIP-TDI liability. So ceralasertib is now in phase 3 trials, and the success of the compound also inspired a number of follow-up approaches. Some of them, I think, nicely exemplify the versatility of the sulfoximine group in drug discovery. So you see here an example from a Chinese biotech. So they basically utilized the second alpha position to come up with a series of bicyclic anylated sulfoximine derivatives, which displayed very good in vitro potency. The next example is another ATR inhibitor, and this is the clinical ATR inhibitor from Artios Pharma. This, to my knowledge, is the latest clinical candidate which contains a sulfoximine group disclosed in the literature. And it's, I think, also the only example which is not an NH sulfoximine, but where the sulfoximine nitrogen position is utilized. So they basically inverted the sulfoximine moiety, so that now the sulfoximine nitrogen is directly attached to the central scaffold. They also worked on this second exit vector to identify Artius 380, which shows again very high aqueous solubility, good potency, and also a favorable human PK profile to support a good therapeutic index of an ADR inhibitor. So this compound is now in clinical phase 2 trials. So at Bayer, after failing with pan-CDK inhibitors, we turned our attention to the selective inhibition of exclusively transcription-regulating CDK9. The starting point of this program was in-licensed from the Lead Discovery Center in Dortmund. We were attracted to the compounds by the good activity against the target, but mainly by the very high kinase selectivity, including the family of CDKs. On the downside, the starting point revealed low aqueous solubility and very moderate permeability, as well as high efflux, overall resulting in low oral bioavailability in species. So in a full-blown lead optimization program evaluating all molecular positions, we finally selected ataviziclib as our clinical candidate. The compound shows comparable potency and selectivity, but significantly increased aqueous solubility. Nevertheless, the compound shows further improved permeability and reduced efflux, resulting in significantly increased oral bioavailability. Moreover, the switch from the sulfonamides to the sulfoximine removed the potential ZIP induction liability. Ataviziclib also revealed promising PK in humans upon oral administration, but daily application of this selective CDK9 inhibitor resulted in neutropenia as a dose-limiting toxicity. In a follow-up program, we investigated if the switch from daily oral administration to once-weekly IV administration could lead to a better therapeutic index. Solubility was a key lead optimization parameter in this IV program, and we again evaluated a broad variety of polar solubility-inducing functional groups at this position here, which is directed to the exit of the ATP binding pocket. But none of these alternative compounds revealed an improved overall profile. So finally, we decided to stick to the sulfoximine moiety at this position, and a number of scaffold hops led to the identification of inhibition. Compound shows the significantly increased biochemical and cellular activity, as well as significantly increased cell activity, and aqueous solubility is sufficient to enable the formulation of the human dose for once-weekly IV administration. Inhibition recently demonstrated early signs of clinical efficacy in patients with advanced malignancies. So at the end of the inhibition approach, we got some additional time to look into potential IP gaps, and we became interested in the idea to stabilize the bioactive conformation of our inhibitors by macrocyclization. This rapidly led to a novel series of macrocyclic CDK9 inhibitors, which displayed significantly increased cellular potencies. Moreover, the compounds had a differentiated PK profile with further reduced clearance, significantly increased volume of distribution, leading to much longer half-lives. And also in SPR experiments, we recorded much longer target residence times for the macrocycles, mainly going back to their increased affinity to the target. So the biochemical value you see here is basically just reflecting the assay wall. So based on the significantly increased cellular potency, especially against certain sensitive cell lines, we also evaluated the macrocyclic CDK9 inhibitors as payloads for ADC approaches. And here we took advantage of the sulfoximine nitrogen position, which offered the rapid introduction of various linkers to the antibody with high chemical diversity at this position. On the next slide, you see a more recent example. in which the sulfoximine and the sulfoximine function was introduced to a natural product to allow conjugation approaches. So the amanitin is a very deadly toxin. It's found in the amatoxins, so in mushrooms like the death cap and the destroying angel. So I guess, the names already indicate that there is a certain potential as the payloads for ADC approaches. And indeed, amatoxins have been evaluated as payloads for ADCs. So the group of paran now switched from the sulfoxide at this position to the corresponding sulfoximine and sulfoximine to allow conjugation via the sulfoximine nitrogen or the sulfoximine nitrogen position. And according to paran, the program has been licensed to an undisclosed industrial partner now. So along those lines, the sulfoximine nitrogen position has also been successfully utilized for the construction of PROTACs. I think this here is the first example, which comes from a group at the University of Nanjing. They identified very potent CDK9 degraders. So they utilized atoveziklib as a starting point, and since the sulfoximine nitrogen is directed to the exit of the ATP binding pocket, it can be easily functionalized with various linkers to the E3 ligase binding group for the construction of the PROTACs. So BO3, for instance, completely degraded CDK9 in cells. The PROTACs still displayed high kinase selectivity, but enhanced antiproliferative activity and apoptosis induction as compared to atoveziklib. And the compound also induced CDK9 degradation in vivo. You see here a second example from a different group at the University of Nanjing, who recently reported very potent ATR degraders. So they started with the serolazatab, and again, the sulfoximine nitrogen is positioned at the exit of the binding pocket, and therefore allows rapid introduction of various linkers to the E3 ligase binding group. So their top compound also revealed selective and effective ATR degradation. It had a reasonable PK profile, which allowed for anti-tumor efficacy testing in mouse models. And here the compound revealed efficacy in monotherapy and in combination with cisplatin. So we've seen now several examples in which a sulfonamide, a sulfone, or a sulfoxide was replaced by a sulfoximine. But what about the replacement of non-sulfur-based functional groups? So amines, for instance, are exemplified in many marketed drugs, but then again, amines can also turn out to be problematic in lead optimization, for instance, with respect to hERG activity and cardiotoxicity, or lysosomal trapping. So based on the rationale that under physiological conditions, most amines are predominantly protonated and therefore tetrahedral, we investigated the switch from amines to sulfoximines in a number of marketed drugs and advanced clinical candidates. So the sulfoximine analog of vardenafil, for instance, was shown to be at least equipotent in vitro. Compound shows reduced lipophilicity, but this does not translate into improved aqueous solubility. However, the compound does show reduced turnover in hepatocytes in vitro. Vardenafil has high permeability and no efflux in the CACO2 assay, whereas the NH sulfoximine analog revealed low permeability and very high efflux. So the extent of this change with respect to permeability properties was surprising for us in the light of the promising permeability properties that we, for instance, recorded with our CDK inhibitors, but Gnam and Bohm independently also reported that, of course, the introduction of a very polar group like the sulfoximine group can result in impaired permeability. So against this background, we wondered if cyclic sulfoximines could combine the promising permeability and metabolic stability with high, with good aqueous solubility. So based on a short synthetic approach that we introduced for the construction of 5 to 7 membered cyclic sulfoximines, we compared the in vitro properties of roniciclib with an ethyl analog and cycloroniciclib. So in line with our expectations, the N-ethyl derivative showed significantly increased permeability and reduced efflux. But the compound also showed significantly reduced metabolic stability in vitro and the trend for decreased aqueous solubility. Cycloroniciclib, in contrast, revealed a trend for improved potency, but comparable metabolic stability as roniciclib. Moreover, the compound revealed improved permeability and reduced efflux, as well as more or less comparable aqueous solubility. So here you see another example of a clinical candidate, which was recently disclosed in the literature. It's the helicase primase inhibitor IM250 from Innovative Molecules. So helicase primase inhibition is a novel mode of action for the treatment of herpes simplex virus infections, which are discussed for Alzheimer's disease. So the clinical candidate pitilivir revealed very good in vitro potency, but development of the compound was hampered by carbonic anhydrase off-target activity, as well as aromatic sulfonamide-related toxicities. And the compound also displayed a rather low CNS penetration. So at Innovative Molecules, they utilized pitilivir as a starting point. They switched from the pyridyl to the difluorophenol and from the sulfonamides to the sulfoximine. This led to a significant increase of lipophilicity, but a reduction of polar surface area. And IM250 then displayed a much improved CNS penetration. Moreover, the compound has a very favorable PK profile with a long half-life, low clearance, high exposure, and good oral bioavailability, so that IM250 also revealed superior in vivo activity as compared to pitilivir in animal models, and the compound is now being evaluated in the clinic. So here is another clinical candidate which was recently disclosed. It's the CoRES complex inhibitor TNG260 from Tango Therapeutics. So to my knowledge, there is not a scientific paper yet which describes the discovery story of this compound, so that I can't really provide a good rationale why they selected the sulfoximine at Tango as the clinical candidate. However, the compound does display very promising efficacy in suitable animal models in combination with anti-PD-1 and is now being evaluated in clinical trials. So the next example, again, shows the utilization of the sulfoximine nitrogen position for the construction of novel modalities. So click chemistry, especially the alkyne azide cycloaddition reaction, has a high potential in biological sciences, but has been kind of hampered by the need for copper catalysis. So this led to the development of alternative click reagents like TMTH, which rely on a strained triple bond, which does not require copper catalysis anymore. However, this compound does not really offer a suitable exit vector. So at Crystal Therapeutics, they investigated the switch from the thioether to the NH-sulfoximine, and this novel click reagent combines great reactivity with small size and low hydrophobicity. Moreover, the reagent can be conveniently functionalized via the sulfoximine nitrogen position with high chemical diversity, allowing for attachment of salts and biologics. So in the paper, you find several examples, several applications, including, for instance, this folic acid functionalized micellar nanoparticle. So this brings me to my last example, which is a novel series of covalent Werner inhibitors from Fox Therapeutics, exemplified here by the compound Fox 1589. So Vividion 214 is a clinical covalent Werner inhibitor. The compound relies on a very interesting mode of action, since in contrast to the clinical competitors, it utilizes an ATP cooperative binding mode. However, the compound utilizes an unsaturated sulfone as the warhead, which in our experience can often lead to high reactivity. So high reactivity warheads can turn out to have off-target activities and additional toxicities. Moreover, high reactivity warheads can also lead to increased metabolism, leading to reduced half-life and low oral bioavailability, as well as potential immune reactions. So against this backdrop, we wondered if unsaturated sulfoximines, which have, to my knowledge, not been described in the literature for covalent inhibition before, could lead to an improved overall profile. So you see here the direct comparison, 214 versus 1589. Both compounds in vitro show more or less equipotent activity against sensitive cell lines like HCT 116, whereas there is no significant activity against insensitive cell lines like SW 620. 1589 shows significantly reduced log-D. To compare the reactivities of covalent inhibitors at Forks, we routinely evaluate the compounds with respect to human plasma stability. And in this assay, 1589 revealed significantly longer half-life as compared to 214. In subsequent mouse PK studies head-to-head, 1589 revealed a longer half-life, reduced clearance, significantly increased bioavailability, and also significantly higher exposure. So this triggered additional experiments from this novel series of compounds with respect to efficacy and also activity-based protein profiling. But today, unfortunately, I can only disclose the data which was published in our first patent application last week. So with this, I come to the summary. So after the late discovery in 1949, sulfoximines have created only very limited interest in the life sciences for a very long time, and there is still no drug on the market which contains a sulfoxamine group. This is surprising, since sulfoxamines offer a unique combination of properties which are relevant for many medicinal chemistry approaches, like high stability, favorable FISChem, H1 donor acceptor functionality, or structural diversity. Over the last years, we've seen significant progress in the synthetic methodology and commercial availability. Moreover, we have a much improved understanding of the properties of sulfoxamines from the medicinal chemist's perspective. So we have seen several examples for the exchange of sulfonamides, sulfones, and sulfoxides for sulfoxamines, leading to a number of clinical candidates. But I guess you can also consider the replacement of non-sulfur-based functional groups for sulfoxamines, for instance, for alcohols and amines. Over the last couple of years, several novel clinical candidates have been disclosed in the literature. For instance, the ATR inhibitor from Artios, the CoRES complex inhibitor from Tango, and the helicase primase inhibitor from Innovative Molecules. But the main interest has been on NH-sulfoxamine so far. However, I hope I have convinced you that the sulfoxamine nitrogen position is a very interesting position for drug designers, which enables the rapid construction of complex compounds, novel modalities, or simply for further tuning of the properties. So with this, I would like to highlight my former colleagues from Schering at Bayer for their contributions to the CDK9 and pan-CDK inhibitor approaches. Moreover, I would highlight Alberto Savent and Vivian Cybulski for their contributions to the general understanding of the properties of sulfoxamines in drug discovery. And I would also like to highlight the Werner inhibitor project team at Forks Therapeutics. So thanks a lot for dialing in, and happy to discuss more. Thank you.
Yeah, thank you so much, Uli. That was such a great presentation. I mean, really, you gave us the roadshow of the sulfoxamines that have been introduced into the clinic. So really spectacular. And we had a lot of audience questions already. So why don't we just directly dive in? It's like
Sure.
really, people are really curious about one thing. For example, you mentioned the chirality of sulfoxamines, right? So
Yeah.
I'm wondering, is there a huge difference between the enantiomers in terms of potency and maybe even also in terms of PK properties?
Yeah. So
Question.
I guess it depends very much at which position you utilize it. I mean, maybe you have noticed that, for instance, in our pan-CDK inhibitor approach, the first candidate was a R and the second candidate was an S. So this is, I think, due to the binding mode. So here, the sulfoxamine group is more or less directed to the exit of the ATP binding pocket. So here, the chirality does not really seem to play a big role with respect to potency. So if you look at the two enantiomers, they were basically more or less very similar in vitro. We did notice some minor differences with respect to PK. So this was in the end also the selection criteria for the two compounds. So here the R enantiomer showed a trend for slightly better PK in animals, and here it was the S enantiomer, but it was really very close, right? So here the chirality does not really play a big role, I would say, but for other programs it does. Yeah. So you have other programs. So for the Werner inhibitor approach, for instance, it does play a big role. So there we have four stereoisomers, right? And they do differ very much with respect to their properties based on the different chirality also, not only at this position here, but also at the sulfoxamine position.
Yeah, Matthew Pfeiffe noticed that actually that change from the R to S, and he was wondering.
Yeah.
So while we're on the slide, Matthew Pfeiffe has another question. How does the hotness of the vinyl sulfoxime compare to the vinyl sulfone? You talked to this.
This is unfortunately what I can't disclose today. So this is, I can only disclose today the data which was published in our first patent application. The additional results, unfortunately, I did not get permission to show today. So I will have to leave it like this here. So patent was just published last week, so we can't really disclose everything right now.
Absolutely. No, super impressive. And yeah, thank you for having this super low latency to already bring this example into the flash talk. We really appreciate it. Another question, Oli, is from Kevin Fody. So he's wondering, among other things, do you think that generative AI would have come up with the sulfoxamine group back then? I mean, he's mentioning sulfoxamines have been in agricultural products. I mean, you know the history very well. So do you think, like, with only where sulfoxamines have been super underutilized, do you think that's something that generative AI would have come up with, or is this all speculation?
Yeah, I guess it's speculation. I mean, to be honest, I don't know, right? I mean, when we started with this, basically, but this was, more than 20 years ago, and I guess the tools were much different. So at that time, the recommendation was not to do it because it wouldn't work. So we made the model compound anyway, and it worked. But I don't really have good guess what AI would do with this. But of course, I mean, now the group is very much established, so I guess it is also something that is picked up from AI approaches, I would guess. But I don't have any hands-on experience with this.
Yeah, makes sense. Yeah, thank you for that explanation. One thing I think both Kevin is wondering, Kevin and I think Alexi are wondering about are there in general any like detrimental issues that are known preclinically? May that be a unique metabolite of sulfoxamines, or typically any like safety issues that you see come up with those in in preclinical safety panels, like the safety screen 87 that you would run? Any like experimental things?
Yeah. I mean, we and others have not really identified a general flaw with sulfoxamine, so I guess it always depends on the overall molecule in the end, but there is no, I think, intrinsic flaw with the sulfoxamine group. As I mentioned, the one concern was that they might be hydrolyzed right away or metabolized right away. This is also something that we did not spot in the clinical programs I was involved with. However, we had at Bayer one example I have in the backup. So this is an example from a program which was led by my colleague Wolfgang Schwede. This is the program that led to the identification of the clinical candidate vilaprisan, which is a sulfone. However, during lead optimization, they also looked into the sulfoxamine analog based on the limited aqueous solubility of the compound. I mean, in the end, this didn't play a role because it's a very potent compound with a very low dose. But they looked into this, and indeed, the sulfoxamine analog again was much more soluble. But in this case, the compound was also 10 times less potent. And still, they compared both compounds in vivo, and interestingly, both compounds basically displayed a similar efficacy at the same dose, which was a bit weird because, yeah, the sulfoxamine was 10 times less potent, but analysis of the blood samples then revealed that in this case, indeed, the sulfoxamine is a bit more like a prodrug for the sulfone. So here, a large part of the dose was metabolized in mice to give the corresponding sulfones. But this is the only example I'm aware of. So for the CDK inhibitor approaches, we did not see a large formation of the sulfone. We saw a bit of this, but it was always like a minor metabolite which was formed.
Yeah, super interesting. Yeah, thanks for sharing that. Patty Melson is wondering here about kind of the geometry and the potential for metal coordination. So you presented, it could act as a sulfonamide bioisoster. He's wondering if it could also act as a potentially stable mimic of otherwise unstable hemiaminals, and is wondering if that could have applications, for example, with histone demethylase inhibitors like KDM inhibitors, where oftentimes you need, really acidic functional groups to.
Yeah. So I mean, in the literature, you find examples, I think, where sulfoxamines have also been utilized as transition state analogs. So due to the tetrahedral geometry of the compound. And yeah, I mean, they are described as that they have metal coordinating properties because they are also used in addition to or prior to the life sciences. They have also been used in ligands for metal catalysis, right? So they were able to complex different types of metals. So indeed, I mean, in our case, they are not able, or in the CDK inhibitor program, they were not able to complex a zinc cation, which was crucial for the carbonic anhydrase off-target activity. So this goes back to an interaction of the sulfonamide group with a zinc cation in the carbonic anhydrase binding pocket. Right. But in general, yeah, I think it's something that you indeed can consider because they are in general able to complex metals.
Yeah, totally. No, that's definitely great to know. Another question here, and I think you kind of teasered that already at your summary slide, is about sulfoxamines in the CNS area. People are wondering about kind of the brain penetration of compounds that contain sulfoxamines.
Yeah, I mean, let's see. So this is basically the only example I'm aware of. Is the IM250. So this is a good example for very high brain penetration, even though it's an NH sulfoximines. So yeah, this is a very positive example. But then again, of course, you have other examples I've shown where permeability was low, efflux was very high, which wouldn't support CNS penetration. It's a bit difficult to click through. Let's see. Here it is. Okay. So yeah, this one, I mean, this is quite remarkable, the difference. I mean, of course, these are two modifications, but still leading to a significant difference in CNS penetration. Again, I think this is some probably you need to try out, at least with a model compound or something, if it would work in your program. But in general, I think this example shows that you can utilize them for CNS targets as well. I mean, one way to improve permeability is, as I said, the functionalization here, often leading to reduced metabolic stability. So therefore also, yeah, cyclization approaches might be also something for CNS approaches, because then you get also rid of the hydrogen bond donor at this position. But in principle, I think this is something you can also evaluate. But yeah, the examples in the literature, I think, are limited.
Yeah, makes total sense. And then maybe with respect to the biological activity of some precursors, you mentioned, I think in the natural product example, the sulfoximines. Generally do people look into the biological activity of those compounds, or do they tend to be like somewhat unstable?
That's a good question. I mean, Christian Gnam just published a review article on this, so he is probably the better source for this. We utilized the sulfoximine function in an early kinase inhibitor program, because at that time it was kind of a intermediate compound. So at that time we built them up by first immunation and then oxidation to the sulfoximine, and we realized that the sulfoximines also had biological activity, but we did not really follow up on this a lot. There were kind of concerns about metabolic stability, hydrolytic stability, but you find this in this recent review article from Christian Gnam in all detail, I would say.
Sounds good. Yeah, Oli, honestly, we have over 20 questions that I think we could go on for an additional hour here with just a question, but I think we would need to wrap it up. So maybe one last, or maybe 1 to 2 last question. One, people are wondering are N-acyl sulfonimines stable in the plasma? And have people thought about kind of maybe pro-drugging, if you will, sulfoximine?
Yeah, I mean, that's also an interesting question. There is an early paper from Bohm. They have been promoting this also as a amide bioisoster for peptides. In my experience, but this is rather limited, the acyl ones were not that stable. But again, this is based on the CDK inhibitor program. But there you saw quite a tendency, which I think you can also utilize for different approaches with respect to stability. If you look at the, let's say, amide-like type structures, the carbamate structures, and the urea structures, or the sulfonamide structures. So depending what you put on here, if it's an acyl, or if you form a carbamate, urea, or a sulfonamide, you can also induce different stabilities with respect to hydrolysis at this position, and also with respect to metabolic stability. So I think this is another advantage, that you can really diversely functionalize this position here, and with this also induce different types of stabilities or potential release mechanisms as well.
Yeah. Super cool, super cool. And then maybe only the last question. You mentioned a few synthetic methodologies to synthesize them, and how they have changed the game and increased, I guess, the usage of the group. Is there a way that you're aware of that you can make them enantioselectively selectively?
Yeah, in principle, you can utilize the Bohm method for this. Yeah, so if you build up the sulfoxide enantioselectively, then basically this method keeps the stereo information. So this is also what we did at Neuroni clip approach. So we build up this enantiomer selectively with a metal catalyzed reaction, and then kept this chiral information with this method here. This is also stated in the paper. So this is one way really to build them up enantiomer selectively. But you have to go via the chiral sulfoxide, to my knowledge. I think there are recent approaches where you can also go the other way around, so that you first introduce the imine and then you oxidize, also keeping chiral information. But this is the method which has been around for 20 years. So you can do this,
Yeah, sounds great. Yeah, Uli, thank you so much for an incredible talk, and you really thank you for staying here to answer all the audience questions. I think
Sure.
people got a lot from it. But again, we didn't get through through all of them, but you gave phenomenal answers, and again, showing the low latency, showing the Werner inhibitor that was just patented last week is something really super cool. So thank you again for that, Uli.
Yeah, thanks a lot for the invitation, and thanks a lot for dialing in, everybody. Thanks.
Yeah, sounds good. And we'll see each other next week at the next Drug Hunter Flash Talk with Bradley Morgan, and he will be talking about the discovery of omecamtiv and aficamten, and those are two cardiac myosin modulators. So stay tuned for that, and we will see you next week. Bye-bye.
Thank you.